fibrous extracellular matrices. And of course, this is a solid mechanics talk, so the focus is going to be solid mechanics. In particular, what are the new mechanics concepts you can learn from these interactions of cells and matrices. And in particular, they're absolutely relevant for two diseases, fibrosis and cancer. And this work is supported by an R01 that I'm a PI on with a clinical person who does liver fibrosis. And this is a new physical science of oncology center on cancer that just started at Penn this Monday, so it's a few days old. So none of the work will be related to that, and it's a little bit away from this topic. We're doing other aspects of cancer, and I was hoping to see Dennis Carter. Apparently he's not here, so this is a grant from Dennis Carter's biomechanics program. So the outline of my talk is, of course, I'll start with explaining why mechanical stiffness is important in fibrosis and cancer. But of course, if you want to do mechanics, you want to reduce this to a simple, well-defined problem that you can solve. Okay, so the problem we're going to ask ourselves is, if you have this is work from Jan Lippard's lab, I'll come to it in a second. If you have a cell or a collection of cells that is sitting in an extracellular matrix, how do we cell remodel the matrix? Meaning, how does it grab onto the fibers and how does it change the overall structure of that matrix? Okay, and how does that remodeling influence the properties of the cells? And this is really important, as I'm going to show you in cancer metastasis, fibrosis, and also in wound healing. Okay, so that's fine. But then we want to ask and define a question. So the question we're going to define is really, really simple. If a cell is going to exert forces, how far can they sense? And how are these sensing, if I'm sitting and pulling here, how far will you feel? Okay, and how are these distances influenced by cell shape, contractility, and the properties of the matrix? So I want to put this in a little bit of a context. So the whole field of solid mechanics started off by, arguably by the solution of the Eshelby problem, right? You have an inclusion sitting in a matrix. The inclusion has got a different thermal mismatch, so it's exerting stresses. How do these, what are the stress fields? So what I'm going to show you is the Eshelby problem in a fibrous matrix. Okay, so define it very, very clearly. Okay, so it's the Eshelby problem in a highly heterogeneous fibrous network. So that's precisely what it is. So we can define it very, very clearly. So this will be the first part of my talk, so of course we'll solve the Eshelby problem. Then we will look at the feedback between the cells and the matrices. And in the second part of my talk, I'm going to talk about another really important phenomena in mechanics, which is necking. So necking in bars is a standard problem in mechanics of materials. So here we're going to look at a system where you're artificially growing a tissue. In this case, the tissue is held between the posts. Here it's held between flexible posts. And so the question we want to answer is this tissue stable? In other words, what I'm going to show you is that if you just hold a tissue between two posts that have some stiffness, the tissue can automatically neck. Okay. This is really important in regenerative medicine because you're going to grow these tissues in some environment, then you need to implant it within the body. So you really need to focus on the mechanics issues. So I'm going to show you is there is a new necking phenomena that has no analogs in non-living matter. So it simply comes because of the coupling between things like stress biochemistry that gives rise to these forces. And I'll talk about implications for tissue engineering, wound healing, and morphogenesis. So starting, as I said, we're going to, the things we're interested in fibrosis, which is excess and abnormal extracellular matrix deposition in response to injury. And this is responsible for 45% of the deaths in the developed world. It's from this article. The reason is things like heart attacks, vascular, kidney, lung, liver, joint, all of this can be attributed to the fact that there is more matrix that's produced. Of course, everybody knows about cancer and cancer too. There are important mechanics issues. So let's so again, to motivate this, let's see how mechanics is related to disease. So here, what I'm showing you are the stiffnesses of liver, lung, and the arteries. So this is a normal liver. The stiffness of the normal liver is about 0.4 kPa in the blue here. The stiffness of a fibrotic liver is 22 kPa. So you see that there is more than 2 orders, nearly 1 to 2 orders of magnitude increase in stiffness during disease. Same thing in the lung. If you look at the stiffness for an uninjured and injured lung, which is fibrotic, there's an order of magnitude increase in stiffness. Same with the vasculature. Okay. So clearly, the disease state is associated. The signature of the disease is stiffness, and this is the clinician that I work with. It's clear from clinical practice. When you think something is stiff, typically it's fibrotic. All right, now let's go to cancer. So typically, a lot of the cancers, here I'm showing you cancer of the bile duct. So this is a bile duct, where the tumor. And surrounding the tumor, there is a lot of extracellular matrix or fibers, as I'm showing here. So these are the epithelial cells of the cancer. This is the fibrous matrix surrounding it. And of course, there are other cells in the matrix. And this is the cancer of the abdomen. You see, these are these cancer cells, and surrounding it, there's a matrix. And what Val Weaver showed some years ago is that if you look at the stroma that surrounds the, this region that surrounds the tumor, its stiffness around a tumor is much more than the stiffness in a normal tissue. Here is 200 pascals. Here it is 2,500. So clearly, it's an order of magnitude difference in the stiffness. Okay. So simply by looking at the stiffness, it's clear that both these disease states are associated with stiffness. All right. So the simple question to ask is why. All right. So what makes the tissue stiff? So a number of different things. If you look at a tissue, typically you got the cells and you got this fibrous matrix that surrounds it. So this is the normal stroma that surrounds in a tissue. And this is the stroma that surrounds tumor. This is work in collaboration with A.P. Kukierman, who's at the Fox Chase Cancer Center, which is about 6 miles northwest of our campus. And what she has actually shown is that as the tumor develops, you get an increase in stiffness as well as the organization of the cells in the matrix. If you look at the matrix here, it is quite random. Here, the matrix is all aligned. And if you look at the cells, they are all more or less rounded. Here, the cells are much more elongated. So the cells are pulling on the matrix, reorienting the matrix, and the matrix is also influencing the cells because the cells have changed their shape. And cell shape is quite an indicator of the amount of force it can exert and so forth, as I'll come to in a second. So the simple 2 questions we want to ask, as I said earlier, is how do cells remodel, reorient the matrix? How does remodeling influence cell shape? Contractility meaning force, and the phenotype. Okay, I'll come to these in a second. So let me start with a few experiments. Because experiments are a good way of motivating this. So this is work. Sorry, going a little too fast. Okay. So this is work we have done with my colleague Paul Jamney, who I understand gave a seminar here maybe less than 6 months ago. So what Paul does is he has a stiff nylon mesh here, and in these nylon mesh, he seeds collagen gels along with cells. So this is this is the collagen matrix before you put the cells in. And you see that the fibers in this matrix are uniformly distributed. So the distribution of fibers is random. Now what he has created here are squares and rectangles too of different sizes. Okay. So if I put a cell in the middle of this matrix and ask yourself, how big should the rectangle be before it forgets about the fact that there is a boundary? Okay. So if the boundary is closed, then of course the cell will feel the boundary. You increase the boundary length, it won't feel the boundary. At some point you'll stop feeling it. Okay. So if the cell size are about 20 microns, if you've taken a course in linear elasticity and the strains are few percent, what's the answer? There is this theorem that goes by Saint Venant's theorem, right? So elasticity has no length scale in it. So you should get a length that is comparable to the size of the cell. Okay. But what Paul finds is that even in if you look at the distribution, here is 200 microns by 200 microns. There's a big change in this thing, and 500 still. So 200 still is 10 times the cell size. 500 is 25 times the cell size. So cells are sensing boundaries 10 times their size or more. So that's a surprising result, right? That requires, it's a good problem in mechanics. Where do you get this extra length scale? Now the size of the cell strain field also depends on the shape. So if you look at non-invasive cells, non-invasive cancer cells, and so here this is work by Ben Fabry's group. So what they do is they actually put these cells in a collagen, three-dimensional collagen gel, and these are the displacement fields. So when the cells are more round and non-invasive, you see that the displacement fields here are 0.6 microns. That's the scale here. Here it is 4.3. So clearly the ones that are elongated, these are the invasive ones, you see they're elongated. The non-invasive ones are more or less around all the time, and their strain fields are more localized. So again, if you look at the cumulative probability and so forth, you see that the sensing distances are again 250 microns here, 400 microns here. Again, so clearly it depends on cell shape and is also an order of magnitude more. Okay. So now let me show you some really nice work from Jan's lab. So what Jan does is, again, it's a collagen gel here, and then he plates epithelial cells here from mammary acini, and so as a movie here from the paper, yeah, the movie is right here. So you see that with time, the cells totally remodel the matrix. So here the collagen is fluorescently labeled. So as the as the system evolves, you see that there are very strong lines that are formed by these cells in the matrix. So they fully reorient the matrix simply by pulling on it. And if you look at the distances, again, you can conclude that the displacement fields are 20 times the cell size. Now in the field of biology, when things like that happen, there is always the question, is this pure mechanics, or is it that the cells are sending some signals and these signals are remodeling the matrix, okay? So to actually show that, what Jan in his lab did is make some cuts. So when you make these cuts, you see that these lines go follow the cut, they go to the edge of the cut. I'm going to show you some simulations in a second where you can also do these box cuts. You can completely isolate this cell cluster, and again, these lines disappear. In some cases, the cells just contract like this without the formation of any line. So again, so this is also an interesting observation that says that the conventional continuum models are inadequate to capture the response. So why is this relevant? So this is relevant because so typically in real tumors, these tracts that form between say two tumors, the cells migrate primarily along these tracts. So this is work from Pat Keeley's group at Wisconsin. And what she has shown is that this aligned collagen predicts poor outcome for breast cancer patients. So when you have these aligned tracts, the cell can just metastasize, so they can spread to other organs. Okay, so you really need this alignment for tumor metastasis. And the all this project started about 2.5 years ago, maybe a little bit less than that, when I got to Penn, when a liver surgeon, Becky Wells, she showed me this really cool data that actually showed that in liver fibrosis, when you start developing fibrosis, so these are the portal ducts in the liver, and here this is a cartoon here showing the collagen matrix around it. When you develop when you develop cirrhosis, it's an extreme case of fibrosis. You start forming these bridging lines, collagen lines between these. So it's again very much related to cancer. Okay. So again, you can also see these strong collagen lines in the real histological sample. And in fact, the grant that we're working on is really understanding the details of this process. So clearly, it's important in fibrosis and cancer. So let's kind of pose a mechanics problem. Okay, how far can cells sense? Okay. So here, all I'm showing you is just the SLB solution, just plotted because I give this talk quite a bit for non-mechanics audience. So this is just designed to show the fact that whether the cell is round, it is elongated but pulling isotropically, spherical but pulling anisotropically, or elongated. Still, you don't get long-range force transmission. The range of force transmission is typically V to the 1/3. I mean, that's not surprising at all. Okay. Now the question is, can you use nonlinear models and explain this? Okay. So the simplest type of nonlinear models you can think of are hyperelastic models. All right. So if you take pretty much any hyperelastic model off the shelf and ask the same question, you're not going to get any new result. You're going to get something very, very similar. Reason being that you don't have a length scale in that theory either. It's just you have nonlinear variations in strain. So that's not going to give you a length scale. Okay. So what we want to do is we want to start and try to put some physical insights into this. And the way we've done this is using a kind of three-stage approach. In the first approach, what we're going to do is we're going to assemble the network by assembling each individual fiber at a time, and then look at the mechanics of it. Okay, but that's fine. So then based on that analysis, what we have done is we've developed a new constitutive relation or constitutive model for these fibrous networks. All right, and using those models, we can look at force transmission and actually predict as a function of the anisotropy of the cell and anisotropy of contraction how does force transmission go. And in fact, you can get 20, 30 times the transmission distances right away from there. And then now we have a model of a matrix, and of course you need a model of the cell, put it together. You can have models of tissue. So that'll be the third aspect. So you can go from smaller scales of the fibers all the way to the tissues. So this part is still a work in progress. And I'll show you some of the necking work that I talked about where some simple examples of this are implemented, but there's a lot more work to do. This is the part I'll primarily focus on. All right, so we start with the discrete fiber model. The idea is really, really simple. You go to a box, you start putting filaments in that box. Every time the filament intersects, you cross-link it. The cross-linkers can have any properties. They can be springs, you can put viscoelasticity, they can be flexible, you can do all those sorts of things. And then you keep doing that, then you get a network, okay? So I'm just showing this in 2D. And then you can make it look like the real network. You can also do this in 3D. And now the question is, how does this network behave? First, without the cells, and then with the cells, okay? So so the interesting thing is that things like heterogeneity, fiber realignment, and all that is captured in the model. If a cell is able to,
The bars themselves,
yeah.
Are they rigid or?
Oh yeah, good question. I removed that slide. Sorry, they are semi-flexible. They can both bend and stretch. Okay, so there is a bending stiffness to it. There is a stretching modulus to it. They're not rigid. Yeah, I had an equation there. I took it out. Yeah, thanks for pointing it out. They can both bend and stretch. Okay, very good. So let's start. Let's look at the overall response of this to just uniaxial loading. So what you typically find in this material, in these collagen gels, is that initially the material is very soft, below like say 20 strain of 0.2, and then beyond that the material stiffens. So again, this linear model is a little bit misleading because you need to worry about true stress versus engineering stress and so forth. But anyway, you see a stiffening effect here. And then finally, if you pull it, if you stretch it a lot more, it's of course going to fail. So at the least, we should be able to reproduce this and then maybe go from there. So that's what I'm going to do first. Try to get the overall properties. So here, what I'm going to do is I'm going to take this network and shear it, and you're going to exactly see how the physics of this comes about. So I have a random network here. Let's see. All right, so I'm shearing this. As I'm shearing it, what you see is that initially I started off with something completely random. But here, when you shear it along the direction of principal stretch, you get very, very straight fibers. So the whole thing is remodeled, and along the direction of principal stretch, you get very, very oriented fibers. And in the perpendicular direction, these fibers buckle, okay? So you get a very nonlinear response where all the individual components themselves are linear elastic. So these fibers themselves were just had bending and stretching stiffness, all treated within linear elasticity. And this is what you get when you pull it in tension. It's the same thing. This is completely random. If you look at this, you form these straight lines. By looking at this, it's hard to tell where those came from. So there's a large-scale remodeling and reorientation.
That's a question.
Yeah.
So when we did studies like this,
yeah,
2005, or less, we found that the response was strongly dependent on the properties of the crosslinks.
Absolutely.
Absolutely. Crosslinks is something.
Yeah. If you look at
pin crosslinks.
Yeah, totally. If you look at this paper, the properties do depend on the crosslinks, and I will come to that in a second. I was not planning to do it, but now I will. The main features are exactly the same. It's just that the toe will move back and forth depending on the amount of crosslink. With the rigid crosslink, you get an earlier toe. Flexible crossings, you get a later toe. But the basic phenomena is the same. Okay, I'll address it in a second. Okay. So now you can look at the stress strain curves and you nicely see the toe and the stiffening region. And why does this happen? Okay. So to look at that, a simple thing to do is you have all the filaments and you have the information on all the filaments. So you can go at each filament, you can go to each element and ask yourself how much of it is deforming in bending, how much of it is deforming under tension. So these are the same curves, non-dimensional stresses plotted in a log-log curve. So you see the cusp right here. And right at that point is where the mode of deformation transitions from bending dominated here, the red line, to stretching dominated. So initially these fibers bend. Then as you deform it more, what ends up happening, the fibers reorient, and then the only way you can deform it then is through stretching. So there is a bending to stretching transition. And in fact, right at this cusp, right at this knee, the ratio of the bending to ratio of bending to stretching is roughly 1. So this is a good way of, later when I'm going to present these results of cells, that's a good way to figure out where the border between long range and short range force transmission is, because you form these fibers, okay? So now let's, so it's clear that we are able to capture the overall response. Now let's see the more heterogeneous response when there are cells, okay? So to do that, what we simply do is we take this, we take this system. We start off with all randomly oriented fibers, uniform fiber distribution to start with, and then you put a cell in there, and and you can apply either force boundary conditions or displacement. There's a one-to-one correspondence between them. So what we'll do is simply do displacement for the sake of argument. Apply displacement boundary conditions and see how the forces propagate and the range of force transmission. Okay, so this is a typical simulation. You can see the displacement fields. The cells are contracting, so right around the pole there is a lot of contraction. Far away it's less. And so these are these are 3 different simulations for a circular cell, ellipsoidal cell, and more ellipsoidal. So the initial and final cell volumes are fixed. So again, we are just doing it radially. You can also apply different force distributions, but then just look at the radial case for now. I mean, here you can see more or less somewhat radial alignment. Here, you see that there is longer range force transmission. And the way these things are labeled is red are those filaments that have axial strengths more than 1%. Blue are those filaments that are under compression, so they are buckled. So there is a lot of buckling in the perpendicular direction here, or in the hoop direction here, radial stress propagation. Now with these simulations, you know that when you transition from bending to stretching, that gives an estimate of the range of influence. So now we can go and ask ourselves, what is the range of, what is the zone of influence around a contracting cell? So clearly when the aspect ratio is large and the contraction is large, you can get, in this case, for these simulations, 30 times the cell size, the volume to the 1/3. And so it's clear that the deformation around the cell increases the function of contractile strain and the aspect ratio, okay? We can also look at cell-cell interaction. So here, what I'm trying to plot is the deformation fields for 2 circular cells at 2 different distances. So you see here that you have, when they are spaced by 50 microns, you form this line that Yan saw in his experiments. Whereas when you go to 100 microns, those lines are gone. And similarly, on the other hand, the cells are elongated at 100 microns, still things persist. So clearly the long-range nature of this interaction is coming because of the fact that the cells are actually pulling and making a little rope. And if I hold a rope, if I'm here and you're in Philly, I can still pull, right? But it really depends what the properties of the cells are, okay? If the cells are elongated, they can deform a lot more and there can be longer range force transmission. So here, what I'm just plotting is sort of a phase diagram that shows the function of aspect ratio and separation. What is the minimum contractile force a cell needs to exert to interact? So clearly when the aspect ratio is large, you don't need that much contractile force as when the aspect ratio is small. So clearly, we are able to capture these features in experiments. Yeah.
So I don't understand, what's the property of the network that is between the cells? I mean, how do you choose that?
It's based on how many milligrams per milliliter of collagen it is. I forgot to answer your question, so let me come back to it.
Right.
All right. So in all cases, you will basically get a knee followed by a stiffer region. Okay. So if you change the nature of the crosslink, the knee gets pushed here, and you will get a response like that. If you use softer crosslinks. Okay. If you increase the matrix density, this gets pushed to the other side. The matrix is very, very stiff. You see that the distance between crosslinkers is small, bending is expensive. Bending goes like, it's a fourth. So the point is, if you keep increasing the crosslink density, you will transition to the stretching region right away. So more denser networks will transmit forces over longer distances than softer networks. You can quantify it.
Yeah, but if you don't know the crosslinks, you don't know where you are in that diagram. And that
Oh, this diagram is for rigid crosslinks. I'm confining my attention only to rigid crosslinks. Flexible crosslinks, the same story, you'll get pushed. Okay, I don't want to put too many things in there. So in all these things, the crosslinks are rigid. That's good enough. If the crosslinks are flexible, you just move the transition region. Okay, and these are all experimentally testable because using lysol oxidases and other things, you can change the nature of the crosslink. And I'm sure OV has ways of changing crosslinking in your networks, right? So that's a density you can really play with. Okay, all right, very good. And in fact, it's a fact that tumor cells actually control lysol oxidases. So tumor cells can actually modulate the stiffness around it. So if a cell is tumor-like or invasive or non-invasive, the amount of lysol oxidase it produces is going to be different. So your question is absolutely relevant. We've addressed all of that. But I'm just putting together the basic framework, and it's the same whether there are flexible or rigid clauses. Same story. Just the transitions are different. Okay. So now we have these network simulations. But then what have we learned? So to basically give you a more physical picture, what we thought is we should take the next step and try to do a constitutive model and maybe come up with some real predictions in terms of what the force transmission range is and things like that, in terms of all the properties of the matrices. So that's going to be the next step, the next aspect. Yes.
A and B. What are we?
What's that? Oh, A over B is the aspect ratio. X to
Of what? Of what?
Of the so for here. This A over B is 1. A is this axis, B is that axis. So it's a ratio of the two axes, semi-major and semi-minor axes. So an ellipsoid has got a bigger A over B than a circle. Circle is 1. So it's just a matter of characterizing the shape. Volume is the same, as I said. Yeah.
So when we put the cell in, what happens? How do you solve this problem?
You apply boundary conditions. You contract it. Or you can apply a force. Or you can apply a force either way.
You contract the cell.
Yeah, that's what happens. Cells pull. There's actomyosin things inside and they contract. Yeah.
Another sort of detailed question.
Yeah.
So you say, okay, what's the distance? The strand transmitted. But in the end, of course, there's not like a sharp boundary. So you have to define what is the cutoff strain. Right?
Yeah, absolutely. So the distinction is where do you go from bending to stretching, because that's where you really form the fibers. So when there are fibers, that's really when you transition from one regime to the other regime. So I'm at the knee, and if you're beyond the knee, you have fibers. There's force transmission. Okay, so that's the way we define, and that's really the way they do it experimentally too. Again, you can do a comparison. I'll come to some of the comparisons in a second. Okay. All right. Now let's think about a constitutive model for this. So there are many complicated things you can do, but I'm just going to give you the simplest version that works. Okay. So clearly, you want a hyperelastic model. That's for sure. And so when you take a network like this, you can see two types of filaments. One family of these buccal filaments that are kind of randomly providing a background. And clearly there are some filaments that are aligned. So what we did is we create a strain energy function with the sort of the black lines here, or the bulk strain energy function, which just depends on the invariance, I1, I2, I3, and so forth. So that will give you something of the flavor of a neo-Hookean or something like that. Okay? So that's one component. but that's not really capturing the fibrous contribution in 3D. So to do the fibrous contribution, what we do is we write a strain energy function that depends on the three principal stretches. Okay. So if you take so one is in terms of invariance, the other in terms of the principal stretches. So if you turn the crank, the hyperelasticity crank, the stress that you will get from this fibrous component will be aligned with the direction of the principal strains in the deformed configuration in finite elasticity. So Na tensor Na is basically some aligned along the direction of principal stretch. Okay, and there are three of them. Okay, so then instead of showing you F, I'm going to show you this whole coefficient here. This coefficient here. Sorry. I'm going to show you this coefficient here, which is actually the stress. So if you look at that, again, what we do is, this is a phenomenological thing, and it relates to a lot of the questions that you asked. What we say is that if the strain is below a cutoff, the fibrous stress is 0. I'm plotting stretch here. And then beyond that, you get a hardening region. So there are three parameters here. The first parameter is what is the critical stretch beyond which you're going to get this stiffening. Okay. The second parameter is what is the slope here. So that's a modulus of this material. And then what is the hardening exponent? Just three. And each of these parameters can be related to everything you said. What is the density of crosslinks? What is the nature of the crosslinks? What's the density of the fibers? All of it. Okay. So all the microstructural information is now buried basically in these three parameters. Okay. Now let me show you some really interesting and surprising results with elasticity. Starting with the much simpler problem of just shear loading, and then I will go to HLD. Okay. So let's think about shear loading, and the motivation for this are these really neat experiments by Paul Janvey. So all that he's doing is putting this in a shear rheometer. So just think of, again, there's torsion, but torsion is shear. So you basically take a material and you shear it. Okay. In a linear elastic material, what is a stress-strain relation? It's going to be a straight line. That's exactly what you get in a hydrogel such as polyacrylamide. The shear stress versus shear strain is a straight line. Okay. Whereas in this collagen that is fibrous, you get the nonlinear behavior as well. Yeah, I apologize. So in this nonlinear material, you get the initial lead and then it stiffens in shear. So this is expected. But now let us think about the normal stress. Okay, so in a linear elastic material, if you apply shear stress, you won't get any normal stress. Okay, but in a hyperelastic material, when you apply shear stress, you will get a normal stress. But the normal stress has to be quadratic in the shear strain, because whether I shear it this way or this way, the normal stress should not change sign. This is exactly what you call the pointing effect. So when I teach a course in hyperelasticity, pointing effect is the first new nonlinear phenomenon that you teach. Okay, that is precisely what you get for polyacrylamide. You see, this is much more shallower. It's more of a quadratic fit. Okay, and its sign is positive. If you work Neo-Hookean, the sign will be positive. But in these materials, the normal stress is again quadratic or a more different power law, but it is negative. So you get a negative normal stress under shear. This can only happen for fibrous materials. So if you look at the shear stress versus the normal stress for Neo-Hookean materials, it will look like this. Okay, this is basically textbook exercise. Whereas in our model, you will basically get a normal stress that is negative, has the opposite sign. The reason is that once you form these fibers, they are pulling in that direction. So the normal stress has a different sign than in the hyperelastic material.
So is it tension or is it compression?
It's pulling. It's pulling. So the normal stress is the opposite of what you would get in a hyperelastic material.
I guess so let's say we're shearing is sigma xy, right?
Yeah, you're shearing it like this. So the normal force will be in downward direction.
So you're talking about there's a negative.
Yeah.
It's a tensile stress in the y direction.
No, so the way this is defined is a little bit confusing. So the normal stress is the force that the material is exerting on what's pulling. So the negative normal stress is a little bit of a misnomer. We can talk about this later, but I just so the main point is that you get the opposite sign as in the hyperelastic material. the way Paul wrote it in the first paper, the stress conventions are a little bit confusing, okay? The sign is opposite. It has to do with the fact that you're forming these fibers, and fibers are pulling the thing that you have on the top, okay? All right, so that's one example. The next example is probably much more telling. So here, what I want to do is I want to take a sphere and have a contractile, have it contract, okay? And then what I want to figure out is I want to figure out how the stresses decay in a linear material or a Newtonian material first, and then in this fibrous material with our constitutive law, okay? So if you take a linear elastic material, as I was talking to Bill this afternoon, you will have both radial stresses and hoop stresses, okay? The radial stresses are going to be tensile because if the stress is pulling, you'll get tension in this direction. But the hoop stresses are half the size of the radial stress and have the opposite sign. So if you take the volumetric strain, the stress, there is no volumetric stress, okay? So you take this relation and put it into equations of elasticity, what you end up getting is that the radial stress goes like 1 over r cubed. So that it's behaving like a dipole, okay? Stress dipoles behave like 1 over r cubed. Now let's take our constitutive model and see what you will get. If you look at our constitutive law here, if the stretch is compressive, you do not have any stress. Only when it is tensile, you have sufficiently tensile, you have a stretch. So what is the hoop stress in our problem? 0, because in the hoop direction you are in compression. Okay. So you go back here and put it into the equations of elasticity. You will get a result that the stresses die like 1 over r squared. It's a pretty strong result, right? It's suggesting that the fibrous material is behaving like a monopole as opposed to a dipole. That's because it's really reorienting these fibers, and then reorienting the fibers, the forces are propagated like a monopole, okay? And this clearly depends on the shape, right? Because if I have an ellipsoid, there will be concentrations of the stress at the top and bottom. And also depends on how anisotropically you're pulling. Because that will also, wherever there is concentration of stresses in tension, you'll form fibers. So all of this you can look at in this single slide that I showed, where if you have a radial thing, the range of force transfer increases with shape anisotropy and contraction, and you can make a plot a phase diagram as a function of anisotropy in contraction, which is these arrows, whether they're radial or not, and shape anisotropy. So clearly you see that when things are anisotropic and if there is anisotropy in contraction, there's a long range force transmission. So clearly it's a new paradigm in mechanics where the fibers can reorient and you can propagate forces over much larger distances. Wherever there are concentration of stresses, you will get the alignment. That's why you get much more longer range force transmission. Okay. So again, so this is all numerical. We have some analytical results too, using mass asymptotics and so forth, which is in our paper. I'm happy to discuss it if you have questions. All right. Now, so here is here I'm looking at cell-cell interactions. So here you have two cells in a Neo-Hookean matrix. The cells are doing the same thing, and the linear elastic properties of both this matrix and this matrix are the same. But this is very non-linear, and you see that here, when they are separated by 40 times their size, clearly the cells can interact. And actually, if you go to 2 dimensions, meaning if I had a planar substrate, put a cell on it, it's very well known that there are stress concentration at these edges. So actually, in 2 dimensions, you'll actually propagate forces over much larger distances. This is what we're working on at the end, where you can take the same cell, put it on a 2D fibrous substrate and 3D. You can get different results. Wherever there's stress concentration right there, you're going to get this alignment. Okay. So 2 of the acini in his experiments interact over much larger distances. So it's a lot of interesting mechanics you can do with this. Now, where we are going with this is, so everything we know about cells, forces that cells exert on matrices, is obtained by what's called this traction force microscopy. So what you do is you put a bunch of beads in the in this gel underneath, use linear elasticity to back out what the forces are. Clearly, we know that that doesn't work for collagen and fibrin and other fibrous networks. So we are working with Christian's lab to basically look at develop a traction force microscopy in fibrous networks. So we can actually get the real, much more accurate description of the forces that cells exert. Okay. And now I'm just going to talk about a couple of experiments real quick. So here, what I'm showing you are some experiments from Chris Billier's lab. And so what Chris did is he has a glass slide here, and then he puts a gel on the top. The gel can be polyacrylamide, which is a hydrogel that is non-fibrous, a fibrin gel, or a collagen gel. Okay, so these two are, this is non-fibrous, these two are fibrous. And what's being plotted here is the thickness of this gel versus the spread area of the cells. If you put cells on glass, they spread a lot. If you put cells on a soft substrate, they don't spread as much. So here, as you keep, if the gel thickness is small, then it's basically seeing glass. They will spread quite a bit. As you start increasing the thickness of the gel, the spread area will come down. But you see the big difference between the polyacrylamide and fibrin and collagen. Okay, polyacrylamide gel forgets about the fact that there is glass less than 50 microns, when they are less than 15 microns thick, which means they are basically sensing over a typical cell size. Whereas in collagen and fibrin, it is much more longer range, and you can see that nicely in these simulations. So the same linear elastic properties, the fibrous matrix is there is much more longer range force transmission. Okay, and here are some. This is the theory. Those are some data points that they have. You can, so other beautiful sets of experiments that Yan is working on here is he takes these, these cell clusters and simply makes a cut. And once you make a cut, you can figure out, the cell is going to contract, you can figure out how much gap will develop, okay, because of the contraction. And so these are some simulations, and these are the main results. So you see that if you have a nonlinear material that is non-fibrous, then the gap is small. In the case of fibrous material, the gap is large. And we're working with Yan to really quantify this. So really proving the long-range force transmission. So here, in all of this, how am I doing on time? How many? Yeah, when should I finish? 5:15. Okay, I'll try to wrap it up quickly. So the next thing we are working on now is how do remodeling of cells, how do the remodeling of the matrix influence the cell properties, okay? This is a really, really complex problem that I'm trying to put in this cartoon. So this is the cell membrane, and this is inside of the cell that's outside of the cell, okay? So when the cell is sitting in a matrix and exerting tension, then what that can do, for instance, is pull on these integrin bonds, which are the adhesions. And that can release some signals. And what these signals can do is it can affect some of the molecules that I have shown here, the details of which are not important. But what ends up happening is that you end up binding more myosin, so it pulls harder. Okay, so that's a basic idea. You can look at it through this pathway, or you can look at it through the MLCK calcium pathway. You will get more binding. So clearly, with tension, there is an increase in the motor density, the contractile force. So these details are quite complicated. There's a lot of stuff going on here. But if you're taking a thermodynamic approach, you're kind of viewing this from a macroscopic point of view. The main thing here is that there is a motor density, and then in equilibrium, without the stress, there is rho zero. And simple thing you can say is that there is a mechanocoupling feedback parameter called alpha, okay, which is telling you about all these processes, all of them coupled. Now what we did is we worked actually with Chris Chen. And he has this nice way of holding tissues with different stiffnesses. And these are the posts that are holding the tissue. So I'm not sure I'll have time to talk too much about it, but the point is, if you, with these experiments, we've actually determined what this alpha is. You can, for different cells, you can get what the alpha is. The details are in this article. Now, in the last 5 minutes, I just want to focus on the effects of alpha on the cell shape, cell phenotype, and things like that, which have a lot of relevance for cancer. Okay, so and I'm sure Bill as well as Wei will know some of these things with things like shape transitions of inclusions. So in solid mechanics, that's a big thing. If you put an inclusion in a certain matrix, will that inclusion be spherical or will it be ellipsoidal? So I'm going to give you the bio equivalent of that. Okay, that's what I'm going to do. But it's very, very different. It's completely different because of this chemical signaling. So to do that, of course, so I've been spending a lot of time developing this theory. So you need to have things like the strain energy of the matrix, the the energy of these molecular motors and so forth, the external work, as well as the chemical energy. And in fact, yeah, I won't go into the details. I'll just give you the results. So here are some results where what I'm doing is I'm taking a cell and putting it in matrices of different stiffness. So this is a matrix. This is the stiffness of the matrix, and this is the strength of signaling. Okay, the mechanochemical coupling. So when the mechanochemical coupling is weak, it is below this number in this case, you basically get only the spherical shape. Makes perfect sense. Surface energy for a given volume, the surface energy is minimum for a sphere. But then once you turn this mechanochemical coupling on, what you end up seeing is that you get a first order transition where the cell shape changes from spherical to being elongated. Okay? And so the interesting thing is you can move both in the stiffness space and signaling space by simply using drug treatments. Okay? So treating the cells with different drugs, you're going to change alpha. Stiffness, you can easily change. So, and again, cancer cell may have more signaling strength, and so these parameters may be different for a normal cell and a cancer cell. It's well known that the cancer signaling pathways are different. So can we say anything quantitative about it? So we worked with, this is a work in progress with Etty Kuckerman. And so here, what I'm showing you are matrices that are derived from tumor-associated cells and matrices that are derived from normal tissue. And then you put the same cell in here. So along this axis, what I'm doing is I'm keeping the signaling the same and I'm moving here. So you see that the shapes are more elongated here than there because you're simply stiffer. Okay. You can also go along the signaling axis. How can you do that? So this is Wortmannin. Wortmannin is basically going to affect some of the integrin signaling. So, and this is in a dose-dependent manner. So you add a little bit of Wortmannin, the shape becomes more elongated. You add more, it becomes. So clearly, you can you're keeping the stiffness fixed and you're moving along this axis. So it goes from sphere to elongated. So all these shape transitions you can completely capture with the chemomechanical coupling. Okay. You can do the same thing in 2D as well. So in 2D, the physics is a little different because you don't have the constraint, you don't have the mechanical constraints on top. So basically, a cell that is hemispherical to start with becomes cigar-like. And then it goes back to being pancake-like, but the spread area always increases. It's going to keep spreading more. Absolutely consistent with the data from my colleague Dennis Disher. So I want to finish with these really cool experiments. And these are experiments from Guo et al. And it's a group in Caltech. Now Yan has similar experiments, but here what they did is they took a bunch of these epithelial cells, put it in a collagen gel of this strength. And if you look at what these cells are doing, basically these cell clusters are just moving around in this in this matrix. Okay. Now, if you look at it in this case where you have increased the collagen density, in other words, increasing collagen density means increased stiffness. See, they're completely different behavior. The cells are just coming out; they're disintegrating. With our theory, we can explain this. Okay. So here. Here, what I'm showing you is the change in free energy. When you have a cluster with 101 cells, you basically split a cell, and this side is the aspect ratio of the cell, this is the energy change. The main result is that in very, very stiff gels and soft gels, you don't get the splitting. You need an optimum stiffness for getting the splitting. So it's clear that the cancer cells are doing something. They are really getting biological factors out, they're engineering the stiffness of the matrix and so forth. So it's a very rich area where mechanics really couples with biology, signaling and so forth. And we've just only scratched the surface. There is a lot more to do. So I just want to conclude by saying that starting, what we have shown is we've developed this theory for random fiber networks. And we can really see the formation of these aligned tracks along which things like the cells like cancer cells spread. Okay. We've also developed a 3D convolutional model to explain long-range force transmission. And feedback between matrix remodeling can lead to state changes and a bunch of instability. So Yang's experiments there will be critical. And so one of the things we excluded or we need to include are things like matrix production and degradation. The cells also chew up the matrix. They also produce new matrix. So we really need to include them. We can do that easily in our model, but then we need close coupling with experiments to see what these rules are. And these rules could depend on actually the stress state. Okay, so That's and of course, focal adhesions are key. We've been working on it. I just don't have the time to present this. And so, unfortunately, in the interest of time, I have to skip all about everything that is all the necking stuff. But I'm happy to talk to you about it. So I'll just conclude by thanking my group. So this is a bit of an old picture. You see, Divakar is there. He graduated. So this may be 6 months old. And of course, there are new people in the group. So, of course, this work was done by Abdullah, who's also left the group and currently is working for GE, and Heilong, who's still in the group. And of course, a whole bunch of collaborators, and it's only a partial list, at Penn. So AT, we worked with her on the cell shape. Chris is a collaborator that's pretty much on all our papers. Lou, we've been working on tendon, I didn't present that here. Becky is the person I work with on the liver mechanics. Beth Winkelstein is a person who works on pain, and there's a lot of matrix remodeling issues in pain. So she works on spine pain. So there's a lot of interesting mechanics there. Rob Mock, with whom we've been working on adhesions. Dan Hull is a new faculty member. He does organs on a chip. So that's a cutting-edge area. And Doug Smith, who's been working on traumatic brain injury. So I think I'll stop there.
So I understand when you're using, if you use like the Neo-Hookean model, which is traditionally used, I guess, for conventional polymers, you get a very different response from your model. My question is, micromechanically, what's different? Because you started the talk with this linking model, we have cross links and bending stiffness, and that's my understanding. That's kind of the same thing that's used micromechanically to study polymers. So what is it that's so different about, I guess, these materials?
So it's not really modeling this reorientation. You see, the polymer mechanics is, the Neo-Hookean model is not really accounting for the fact that you get this gross reorientation and remodeling and formation of these fibers. That's what it is ignoring.
Right. But
okay.
So
the point is, it's very anisotropic, right? So the thing is, tension and compression are very different. In a Neo-Hookean model, tension and compression are the same. You don't have this buckling and things like that when you're just doing polymer chains. Whereas when you're doing semi-flexible filaments, tension will orient. The compression will basically buckle. So you have very less stress in compression. Micro buckling is very, very easy. Whereas tension, that's the main difference. So it's not very complicated. You use a different strain energy function. Strain energy function is not written in terms of invariance, but it's written in terms of the principal stretches. And they, in turn, give you a stress in the direction of where you're aligning. That's the primary difference. And that simply gives you the 1 over r squared versus 1 over r cubed, as you see. So it changes the physics a lot. Yes, Peter.
Right, so in your fiber model,
right,
you have an additive decomposition of the energy into a bulk and a fiber part.
Yeah.
And I noticed in your figures that you were choosing the bulk modulus to be 10% of the fiber modulus. What is that bulk? First of all, what is, in your models, what's filling the interfibrillar space?
Nothing. It's just a network. It's just a network.
But the network is sitting in what? A fluid?
Absolutely. Yeah.
So what does that bulk modulus represent? And what happens if you take it down towards 0, for example? First of all, what does it represent? You've chosen 10%.
Yeah. So when you write poroelastic theories, right? You have a property of the matrix, and then you have your water, right? So we are assuming here that there is enough time for water.
So it's in equilibrium.
Yeah, it's in equilibrium. Yeah, there's enough time for water to flow. We are just looking at the elastic properties of the.
But then to take the analogy further, is it drained or undrained? That makes an enormous difference.
It makes a difference. But drained and undrained, it simply depends. It's within an order of magnitude, right? So we are simply trying to capture the main effect of the fibrous thing. You're absolutely right. We have a model where we have poroelastic version of this, where the overall volume is conserved. So whatever is the change in volume here is replaced with. I showed you that article. So we have that, but I don't think that's a detail. That's really a detail. We're trying to understand the mechanics of networks and compare that with mechanics of more.
I understand.
Yeah, you're absolutely right. I mean, in situations where water has enough time to flow, you should be using the drain, not enough time, and in between you should be using something in between. But they're not orders of magnitude different, the drain.
Yeah, right.
Yes.
So in the introduction, you show like a picture of the tumor, and you show like a high alignment of
Yes,
the
yes,
fibers.
yes.
So I'm thinking about like what decides like the direction of the alignment, and also like in your model, if you have a spherical cell, or like do you have a
You get only radial alignment. So in a spherical cell, you'll get radial alignment. But if there's another cell nearby, it will align along that. So the alignment depends on where the other cell is.
Yeah, but that picture shows like all.
Which picture are you talking about?
In the introduction part.
Oh, yeah. So that's actually the stroma right outside the matrix. I can show you more pictures. So what ends up? So that's actually from pancreatic tumor. So what ends up happening is if you have a tumor like this, that is spherical, a spheroid, then you will get alignment radially. So it's one section of that radial alignment, where you're showing that the fibers are aligned and the cells are aligned along with them. So that's basically what that is. Yeah.
Could you explain a little bit more about the cell signaling? You had that thought where
Absolutely.
what kinds of cell signaling? Like
I'll tell you. Can I go into it? Yeah. Okay, so there are two signaling pathways here that I'm showing. There are many others, but let's just focus on, say, this one, for example. So if you take calcium, there are mechanosensitive channels in the cell. So when you put something under tension, there can be more influx of calcium. Okay. So what that will do is it will phosphorylate, this myosin light chain kinase. So upon phosphorylation, more myosin will attach to the actin filaments and pull harder. So simply by applying stretch, you're affecting more contractility. You follow that point? Same thing it's a different incarnation here where you have integrins. And integrin is a mechanosensitive molecule. So when you pull it, it releases this factor called SFX. And what that SFX does downstream of it, it changes this equilibrium between phosphorylated and non-phosphorylated rho, which then in turn will, there are other pathways. There are a whole bunch of other pathways. So answering your question, our field theory is based on strains and motor density which are treated like dipoles. Okay, so the slide that I skipped, we have a field theory that depends on rho ij, which is a dipole tensor, and the strain. Okay, and the coupling comes like this. There is a volumetric part to this, and there is a deviatoric part to it. So this really is the mechanochemical coupling. So I believe the SFX signaling is the deviatoric part, and the calcium signaling is the volumetric part. So you can change each one of them independently. Okay, and that's a detail I skipped. So really we can make connections between signaling pathways and a more macroscopic response. Now if you talk to systems biologists, they will write 100 ODEs for each of these pathways. But the bottom line is, what is the macro description? So what I presented to you is a macro description in 1D based on this single parameter. If you go to 3D and write in terms of tensors, there are two. And each one of them has a physical connection. And this is completely new. I mean, this is the first time anyone is going to try to address it this way.
So, one of the things that happens with these networks is that they are alive. And
what do you mean alive?
well, they are constantly alive.
And it is really good.
They're alive, that they are changing,
Yeah, absolutely.
They are constantly changing, and they're remodeling, and sometimes these stress fibers that you get are the product of alpha proteins starting to walk around.
Absolutely, no question.
And so what leads you to believe or that what you are seeing is the mechanism that?
Excellent question. So how fast do you establish mechanical equilibrium?
No, I presume that is
very quickly. But the answer is that mechanical equilibrium is established very quickly. Okay. Whereas the remodeling and those kind of things take place over slightly larger time scales, like hours. So really, when a cancer cell is...
The cells move pretty fast.
They move like, I don't know, Jan can tell us better. I think they're moving, only hours they move, right? I mean,
Well, so typical time scale for some of these experiments are hours to a day or something like that. But the question you're asking in general is an extremely good one, because any attempt to rigorously separate among biology, chemistry, mechanics is fraught with numerous issues. And there are certain experiments where you can do certain things to the system to convince yourself that one of those three things dominates. And something that Vivek has already shown is in a system where some of these fibers form, if you take a pulse laser, you physically cut the fiber, and then if the fiber vanishes quickly and cells respond quickly to that distant mechanical manipulation of a very small cut in the material, that's strong evidence that in that situation, mechanics is very important. But there's many other things going on, like secreted factors, TGF-beta, and numerous other things.
So that's actually good news for us, because we can write more grants. If you finish it, then that's not good.
But
so you want to keep this going.
But the moment that you put the cell in the matrix,
Right.
okay, and the cell starts to pull, I mean, that's, let's say myosin.
Oh yeah, it takes time. It takes time.
Walking over the actin filament.
Right.
Okay, so that takes time.
It takes time, but then that's actually reasonably fast. It's actually minutes. It's actually minutes.
To become tense.
Absolutely. So in fact, if you take the time-dependent version of this theory, we have 3 time constants, and we can take the time constants. So really simple. So if you go here and take, basically there are 3 sets of parameters in here. So you get 3 time constants. One of them will be the time you mentioned, the time it takes for contractility to build. The other will be a time scale associated with the remodeling. So yeah, you can, the multiple time scales you can build it. You can include that in there, right?
I have one question about in the Campbell plot there was a transition between when there are 2 cells, and what is the transition between both of them? There's a contour plot of like a phase diagram between the cell shape and the transition distance.
Yeah.
The previous one, that one.
Yeah.
I was wondering how do you define that there is no interaction versus when there is interaction, and why is the shape not why is like two lines?
Oh, this line here?
Yeah, how is that defined?
Yeah, it's defined. So for instance, so what we try to see is, are there any filaments under tension right in the middle? That's what we ask.
So do you have any explanation of why?
Oh no, totally. Why it should be a curve like this? It should be a curve like this. Why it's like this? For the set of parameters that we looked at, that was the best fit. Okay.
So it's a sharp transition between one line and the other.
Oh, you mean this corner?
Yes.
It's an approximation. It could very well be. So we spanned a lot of points here. We spanned a lot of points here, and yeah. I don't have an exact answer for why it's a sharp corner. It may very well be that but again, the main point here is that if the cells are separated a large distance, and if they are circular, they won't interact. That interaction will start increasing when you come down. So that's the main thing. That's the main message you can take from here.
I mean, yeah. Thanks.
Okay.
I definitely get the message. I was really curious about why.
Oh yeah.
Transitions.
I don't have a good answer for it. Yeah.
Kink there.
All right. Yeah.
All right. The last question.
I have several questions. On the model which you proposed, continuum model.
Yeah.
So I saw in the slide before that you decompose the energy into volumetric and isochoric part, and
that's only for the bulk part. For the bulk part. Yeah.
So these principal interactions which you use in order to model that reorientation, is that for both parts of the energy?
Absolutely. That's kinematics. So that comes from kinematics. You have.
But how do you? I mean, what is this material which you model in the smeared out way an incompressible material? How do you ensure the incompressibility of it?
It's not incompressible. It is compressible. It's compressible. I mean, if you take the gel and pull it, it's not incompressible. It's only when water comes in, the combination is incompressible.
And that you neglect?
No, we can fit it, right? We can fit it. So there is so if you do poroelasticity, the solid part of it can have a nu, can have a Poisson's ratio, which we can fit to the experiment.
Okay. The other question is that I think infinity of your network.
Yeah.
Is that always 4 in these kind of problems?
It's more. By changing the density, the connectivity will change. So
then your discrete model, I mean, the way of how you put the
Oh yeah, it's what people call it's what people call a Mikado model. You take.
This has only functionality of 4, right? I mean, it can't have.
Oh yeah.
So how do you explain that?
Oh, I mean, this is very standard. So the homogeneous properties of the fiber model is not our thing. You know, people have done this for a long time, and those are well-established results for for the homogeneous case.
Yeah, but I mean, you can, I think, have other kind of discrete networks.
No, so for the discrete networks, yeah, for the discrete networks, what really matters is how many. What is the dimensionless parameter that tells you how many crosslinks are there per unit length? That's what's more important.
The final question is, so your homogenized one,
yeah.
The model, does that account for functionality at all?
It's in these parameters. It's all in the parameters, the 3 parameters. We are characterizing it in terms of 3 parameters. You can relate them to the discrete fiber model, both in 2D and 3D. Yeah.
Okay. All right.
All right.
So let's end. I want to take this opportunity to announce the next seminar speaker in a week is Professor Yang Fu from Harvard University. Somehow it happens that this quarter we all have Ivy League school.
Everybody from the East Coast, right?
Yeah.